Welcome to The Mind, the Cure Protocol for August 23, 2026. No newly verified patient reaching milestone in this pass has reached care, so we will not pretend otherwise. We are following what changed in the evidence, what new research it opens, and what still needs proof before it can help anyone. This protocol is a living loop. People bring the questions that matter. Direct evidence changes the map. The source ledger remembers what we have already seen and checks the original date before calling it new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. Small, open-label, single-arm Phase 2 clinical study, preliminary Nature Communications reports results from the NeoZanHER Phase 2, single-arm open-label trial by investigators testing Zanidatamab, a dual HER2-directed bispecific antibody, as neoadjuvant therapy in early-stage, node-negative HER2+ breast cancer with pathologic complete response, PCR, as the primary endpoint. For people with early-stage, node-negative HER2-positive breast cancer, the question is whether a HER2-targeted approach before surgery can be reliably matched to the patient's most likely. This 20-person, open-label, single-arm Phase 2 study reported a 30% pathological complete response result. It does not establish that Zanidatamab alone outperforms current care. Next, refine patient selection and test safety and long-term outcomes in a controlled study. Preclinical animal study in sheep, not tested in humans. Researchers reporting in Nature Communications showed that an acellular bioengineered regenerative conduit, when given external support, achieved 100% patency at 180 days and remained patent to 18 months as a coronary artery bypass in an ovine model. Unsupported grafts failed early from kinking and thrombosis. For people who need coronary bypass surgery, a ready-made engineered conduit could matter if it can make the jump from animal surgery to human care. The evidence is an ovine study. Supported grafts reached 100% patency at 180 days while unsupported grafts failed early. It is not human clinical evidence. Next, test surgical handling, safety, and durable patency in people against current graft options. First stage, open-label, single-arm Phase 2, no randomized comparator. Researchers at NCT 045433071, reported in Nature Communications, announced that a first-in-human clinical stage of combining CXCR for inhibition, Motixafortide, PD-1 blockade, cemiplimab, and gemcitabine, nab-paclitaxel achieved high partial response rates in treatment-naive metastatic pancreatic ductal adenocarcinoma, supported by single-cell correlatives. For people newly diagnosed with metastatic pancreatic ductal adenocarcinoma, the question is whether adding CXCR4 and PD-1 blockade can materially improve the first treatment path. The first stage was an 11-person, open-label, single-arm study. Its 64% partial response result cannot show how the combination compares with standard gemcitabine and nab-paclitaxel. Next, the ongoing multi-center randomized Phase 2 trial must show whether MCGN performs better than GN, with durable benefit and acceptable safety. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the cure protocol stays current without pretending the work is finished.